Hi, my name is Florian Wagner, and in this video I'm going to try to give a high-level overview of different normalization methods for single-cell RNA-Seq data. So I'll try to explain these methods in an intuitive way, and I'll try to simplify some of these methodological details so that they're a little bit easier to understand. So I also want to add a caveat. This is not a review of the scientific literature. Like I said, I will skip over a lot of details to make things easier to understand. And also, this is just sort of my personal take on, how these methods work and my attempt to explain them in the best possible way, in a simple way. So with that said, I want to talk about what I actually mean by normalization. So normalization is a term that gets used a lot in different contexts, and I just want to define it here, at least for the purposes of this video, as everything that happens from everything that you do to the raw UMI counts that you have when you have a single-cell RNA-Seq data set. Everything you do to that data before you actually run a PCA, because PCA is often the first step that you do for example, for visualizing the data as in a t-SNE or UMAP. So everything that happens here in between is what I refer to as normalization. And also, again, for the purposes of this video, to make things a little bit easier, it's helpful to think of normalization as a two-step procedure, where the first step is a scaling step and the second step is a transformation step. And in papers and publications, these steps are often discussed in combination, but I think it makes things a little bit easier if you think of them as two separate steps, at least for now. So in the first step, the scaling step, that's actually fairly straightforward. Here's an example data set where you have maybe 3,000, a little bit over 3,000 cells, and on the y-axis you have the number of transcripts detected for each cell. So if you sum up all the UMI counts for each gene in each cell, this is the number you get, and you see there's a big variation, big range of values from roughly 2,000 to more than 14,000. And in the scaling step, the idea is to multiply the values, the expression, the UMI counts for each cell by some factor, by a cell-specific factor, so that after you do that, all of the cells now have the same transcript count. So you multiply the small values, the UMI counts from these cells that have few transcripts, you multiply those by some value larger than 1, and let's say you multiply these large values by some value smaller than 1, so that they all even out. And the purpose of that is that it's twofold. So first of all, you often have completely different cell types in a single-cell RNA-Seq experiment, and different cell types can have different amounts of mRNA in them. Some cells have more mRNA, some cells are larger, others have less mRNA, and so the number of transcripts detected it depends on how much RNA is available in the cell to begin with. But we're often not interested in actually comparing these absolute transcript counts because, they are different for different sized cells. Instead, we want to compare concentrations. So that's essentially what you do here. You convert these UMI counts to concentrations. So let's say if you scaled everything to 10,000, now you would have the measurements as how many transcripts are detected per 10,000 transcripts, independently of which cell type you're talking about. And that's often much more useful than comparing absolute counts. The second purpose of this step is even when you have cells from the same cell type, because of technical noise, because of differences in how well the chemistry works inside each droplet, you get a lot of variation even between cells from the same cell type. So that doesn't have to be biological. It can just be that the chemistry worked a little bit more efficiently in one droplet than in the other, and so you get, let's say, twice as many transcripts detected in that droplet. And so the scaling step also gets rid of those differences. So it goes a long way in removing these what's called efficiency noise. So you basically kill two birds with one stone here. You convert to concentrations, and you also get rid of efficiency noise. So that's fairly straightforward. The second step is the transformation step. So that's where we're going to spend most of the time in this video talking about. So I will go over three different methods for this transformation. The first two are what I call simple transformations. So a log transform, which is what being used, for example, in the standard Seurat workflow, or a square root transform. And the alternative to these simple transformations are Pearson residuals, for example, in a method called sctransform. And so simple transformations, the idea is simple. You're basically applying some function independently to each measurement. So I is the index for the cell, j is the index for the gene. So xij is the measurement for the UMI count for gene I, for cell I in gene j. And you're just applying some function f to that measurement, to all of those measurements, and you get some transformed value y, and that's all that you do. So we're gonna, like I said, talk about these two different transforms. But in order to understand, what's different about them and why you might want to choose one over the other, it helps to understand a little bit more about like the noise characteristics of single-cell RNA-seq data. Here's actual data from an actual experiment, which I think is really neat. This is from a publication from 2015. And so in this experiment, the authors, they diluted an mRNA sample into droplets. So each droplet, the idea is that each droplet contains exactly the same mRNA, mix of mRNAs, and they have about 1,000 droplets or so. And so whatever variation you see in the number of transcripts detected between these droplets is presumably purely technical, because they should be identical in terms of their actual in terms of the actual mRNA content. So here's, for example, here's a histogram of the number of transcripts detected for GAPDH, which is a very highly expressed gene. And you see it sort of ranges from maybe 30 to 60 or so. And presumably all of this variation is purely technical. So this experiment allows us to actually measure how much noise there we see in single-cell RNA-seq data. And the basis of this technology from this paper, it's still the same. We're still counting UMIs, and this is still applicable to data generated in 2021. So what's important to understand is that whatever in a real single-cell RNA-seq experiment from some tissue, whatever expression differences we observe, some of those differences are because the cells actually have true expression differences. They actually express different levels of genes. But then they can also be due to technical noise, like we've seen here in this experiment. And often it's not even clear which of these differences, which of these sources contributes more to what you observe. So for many genes, the technical noise might contribute actually maybe a bigger factor in explaining these differences than the true biological differences. It depends on the gene, it depends on the cells that you're comparing, and the expression level of the gene. Okay, so first we're going to look at how these different transformations affect true biological differences. So we're assuming for a second that there is no noise. All of the measurements that we observe are because of true biological differences, and we're just going to ask, how do these transformations affect these true differences? And so for that, we'll have a little toy example here. So imagine you have two cell types, cell type A and B, and two genes, 1 and 2, and both genes have a twofold increased expression in cell type B compared to cell type A. This is all after the scaling, let's assume. But the difference is gene 2 is 100 times more highly expressed than gene 1. And this actually, this happens in real data. There are genes that are 100 times more highly expressed than other genes. So the difference, if we just look at the raw data, the difference between cell type A and B is 1 for gene 1 and 100 for gene 2. So if we just take the raw data and we do a PCA on that, this gene 2, these expression differences in gene 2 would be 100 times more impactful or visible than these differences in gene 1. So what happens if we did a log2 transform to these data? So we imagine we're just taking the log2 of these values. So now we have 0, 1 for gene 1, and 6.64 and 7.64 for gene 2. Now the differences are the same. So after a log transform, fold changes, equal fold changes become equal differences, right? The log changes things from a factor to a constant, basically. And what happens when we take a square root? So when we take a square root, then the differences between gene 1 are 0.41, and the differences for gene 2 are 4.1. So here the differences are still 10 times higher, but it's not as extreme as here where they were 100 times higher. But it's also not as radical as the log transform where now the differences are equal. So the square root is somewhere sort of falls in between the raw data and the log transform in terms of how it affects true expression differences. So like I said, without transformation, the analysis is completely dominated by differences among the most highly expressed genes. And after a log transform, certain fold changes, whatever the expression level is, fold changes remain are constant. And after square root, it's somewhere in between. Okay, so next we are trying to understand how do these different transformations relate to the noise profile of single-cell RNA-Seq data. So I've already talked about that noise is an important factor in single-cell RNA-Seq experiments or data. So let's go back to our GapDH example here. In order to quantify how noisy something is, we need something called the mean and the standard deviation, and something called the signal-to-noise ratio. So for this gene, in this data set, since we're only measuring technical noise in this experiment, the mean is 46, the standard deviation is 7.4, so we can say that the signal-to-noise ratio is the mean divided by the standard deviation, which in this case is 6.2. So the higher the signal-to-noise ratio, the less noisy, the more accurate our measurements are. So in this case, the mean, which is presumably the true expression level of this gene, is 6.2 fold higher than the standard deviation, which is good. So the higher this number, the more accurate. So now we can do this for all the genes in the data set. So GapDH, like I said, is a very highly expressed gene. So on the x-axis here is the square root of the mean expression that we observe. GapDH is one of the most highly expressed genes in this data set. Most of the genes have a much lower expression. This is on the log scale here. And you see that there's this very clear linear relationship between the square root of the mean expression and the signal-to-noise ratio on a log-log scale. So it covers, 2 orders of magnitude, and it's a very clean signal here. So what that means is that for UMI counts, the signal-to-noise ratio depends directly on the square root of the true expression level of a gene. So it's directly proportional to the square root of the true expression level, lambda ij here. So this is true for all single-cell RNA-seq data roughly. It's an approximation that uses, as long as these, as you have UMI counts. So there are some methods, some technologies that don't use UMIs, but for most of 10x data, this holds true approximately. So knowing that the higher the gene is expressed, like if you take the square root of that, that's directly proportional to how accurate you're measuring the expression of that gene. Which of these transforms is sort of the most appropriate or the most fitting for this type of noise profile? Well, I think it's clearly the square root transform, because basically after a square root transform, you assign weight to each measurement roughly proportionally to how accurately you're measuring or that measurement is. And that seems kind of right. That seems kind of logical. So if you have a very accurate measurement, you want to give that more weight than a really noisy measurement. It doesn't make sense to treat really noisy measurements equal to very accurate measurements. And this is kind of a little bit what happens with the log transform. The log transform makes it so that the most highly expressed genes are, can have the same, like in this example, they can have the same fold change as lowly expressed genes. But this gene is measured so much more accurately, and this difference here could be purely due to technical noise. So it kind of makes sense to give them a little bit more weight, but it doesn't make sense to give them 100 times more weight, because we're only measuring it 10 times more accurately, if that makes any sense. So that's why, from a noise, at least from this noise perspective, the square root transform makes a lot of sense for single-cell RNA-seq data. Okay, so we've covered these simple transformations, log transform and square root transform. What about Pearson residuals? So Pearson residuals work very differently from these simple transforms. So Here, you're not actually applying some independent function or a function independently to each measurement. Instead, you're multiplying each measurement by some weight, and this weight is a gene-specific weight. So you're basically picking some number here, and you're multiplying all the expression measurements from one gene with this number, with this weight. So how do you choose this weight? And so for Pearson residuals, this weight is just the inverse of the square root of the mean that you measure for this gene. So basically, if a gene is more highly expressed, it gets divided by a somewhat higher number, or all the measurements get divided by a slightly higher number. So it will get a little bit less weight, or multiply or downweighted a little bit compared to a lowly expressed gene. Okay, so this is a very important difference. So instead of transforming each measurement individually, Pearson residuals apply a weight to all measurements of a gene. And so what this actually does, so aside from, this math, I mean, this is fairly straightforward mathematically, but what does this actually do? So what it actually does is it makes it so that each gene contributes to the analysis according to how much evidence there is that it is non-uniformly expressed. So this is very important. So genes that show evidence that they're not uniformly expressed across all the cells in the data set, the more evidence there is, the more of a weight they get if you apply this method here. So like I said, genes that are only expressed in a very small fraction of cells, for example, they get a very high weight here. Even if they're lowly expressed, they can get a fairly high weight, because if they're only expressed in a small proportion of the cells, that means that this mean is very small, which means that this weight becomes much bigger. So you can see how it's not just about the actual, individual measurement. It's actually about, the mean expression of each gene. So this is a very interesting idea. And let's just see how that plays out. So in some respect, Pearson residuals behave similarly to the square root transform. And what I mean by that is, if we go back to our example, and we're looking at what happens with Pearson residuals, assume for a second that cell type A and B are each 50% in the data, and you just go through the math, and what you find is, again, similar to the square root transform, that after you convert everything to Pearson residuals, the difference for gene 2 is 10 times higher than the difference for gene 1. And that makes sense because you divide by the square root, so what you get is, again, the square root. So it's, in some ways, it's similar to the square root transform. However, in other ways, it's completely different. So it depends totally on the data. So here's an example, a different example now, different toy example, where you again have cell type A and cell type B, each at 50%, but there's two subtypes of cell type B. And subtype 1 is the dominant one, has 48% of all the cells, and subtype 2 is 2% of all the cells. And now we have two genes. Gene 1 is, they're both specific to cell type B, but gene 1 is expressed in all the cells of cell type B in both subtypes, and gene 2 is only expressed in subtype 2, which is the rare subtype. And gene 1 has an expression of 8, and gene 2 has an expression of 4.5. It's really interesting to see what happens with Pearson residuals. So after you go through the math, you calculate the mean, you divide by the square root of the mean. Now, with Pearson residuals, these numbers are 4 and 4, and this gene is now 15. So you see how this swapped. So you went from a gene that was higher here to a gene, and now it's lower here. And actually, if you calculate, the variance and how much each gene contributes in terms of its variance, actually now gene 2 after the Pearson residuals contributes more than gene 1 in this analysis. And so that's just an example to show you that it doesn't just matter what the actual expression level is, it also matters how the distribution of its expression. So with Pearson residuals, lowly expressed genes with very cell type specific expression patterns can contribute more to the analysis than highly expressed genes with broader expression patterns, is exactly this example. And so that can be quite interesting and also useful, because we we have these situations in real data, I think, sometimes where we have a gene that's lowly expressed, but it's still very cell type specific. And even though the measurements might be quite noisy, they're still very informative, because whenever we do see that gene, we can be fairly sure that this cell is subtype 2, not subtype 1, right? So to illustrate that a little bit, let's look at some real data. So this is a PBMC data set that I like to use from 10x Genomics, which has about 10,000 cells. And, we, at least at a coarse resolution, we have a fairly good understanding of which cell types are present in PBMCs. They are NK cells, T cells, B cells, monocytes, myeloid dendritic cells, and plasmacytoid dendritic cells. And then the black things are some doublets, I think. So I've manually annotated these different clusters here. And the gray ones are some sort of outliers. And you can see that after a log transform, this works. Log transform separates these cell types fairly well. There's definitely quite a lot of heterogeneity in the T cell compartment here, also in the B cells. And so it's interesting to see what happens now with different transformations. So now here's a log transform, this square root transform. This is one that I like to use. But it's basically just the square root of x, and then you're just adding, combining that with the square root of x plus 1. So this makes it so that lowly expressed genes get treated a little bit differently than highly expressed genes. But anyway, it's basically just a square root transform. And so in this example, the results are fairly similar. I mean, the clusters, these islands in the T-SNE, they're fairly similar. The B cells you see here, they have a slightly different shape, and I think, again, a lot of that has to do with the fact that the log transform punishes highly expressed genes a lot more than the square root transform. So, the B cells, some of these B cells express some genes very highly, some immunoglobulin genes, I think, so they get treated differently by these transforms. But overall, I mean, the differences aren't immediately, at least there aren't huge differences immediately visible. But with the Pearson transform, it's kind of interesting because so a lot of these clusters look, in general, it still looks fairly similar. The B cells now look quite a bit different, actually, in terms of how they distribute a little bit. Maybe a little bit like in the log transform, I don't know. The myeloid dendritic cell cluster is now very clearly separated. It was a little bit separated in the log transform, but it's not very separated. But there's this whole new cluster, this whole new island here, which maybe you can see a little bit here in the log transform. I don't know if you squint a little bit, but here it's a big difference because it's sort of it's like clearly separated from the rest of the T cells. And these are actually, I looked into what they are, and they're actually CD8 positive naive T cells. The CD4 positive naive T cells are somewhere here. So CD8 positive naive T cells, they form their own cluster after you use Pearson residuals, and that's kind of cool because they are very similar in terms of their overall expression patterns to other naive T cells, and not the only, but like they only express, I think, a couple of genes that are different from the CD4 positive naive T cells, and yet they get separated into their own clusters. And I assume that that's because CD8, even though it's maybe not a super highly expressed gene, CD8A and CD8B, they are fairly cell type specific. They're only expressed in CD8 positive T cells, and maybe with the Pearson residuals, that's enough to separate them out as a cluster. And so I think this is a kind of an interesting example. I think it's fair to say that the differences between log transform and square root transform in general aren't huge, but there's quite a bit of difference in terms of how much highly expressed genes can contribute to differences. So in this case, these B cell clusters are separated a lot by some very highly expressed immunoglobulin genes, and with the log transform, the impact of those genes is mitigated quite a bit. But the Pearson residuals, I think they work completely differently. They completely change the variant structure of the data. There's some modifications that have been proposed where you do some clipping because it can get really extreme. So if you have, for example, I think in this data set there is a contamination with red blood cells. I think it's only a handful of red blood cells, but because they have such a unique expression profile and because they're so rare, they become one of the dominant sources of variation if you just calculate the pure Pearson residuals, because the evidence that these hemoglobin genes and these red blood cells are non-uniformly expressed is so overwhelming compared to any other gene that it just completely drives, the variance in that direction. And maybe that's a little extreme because we're often not interested in populations that, only consist of a few cells. And maybe we don't want these genes to have such an outsized impact. And so there's been some clipping proposed to make sure that the Pearson residuals aren't too high, basically. But so this is, I hope you got like a somewhat intuitive understanding of these different transformations and the Pearson residuals. So to summarize, normalization can be thought of as a scaling step followed by a transformation step. The log and square root are simple transformations. They are applied to each measurement independently, whereas the Pearson residuals are looking at the mean, and as a result, they weigh genes based on how non-uniformly expressed they are. Very important about single-cell RNA-Seq data is to know that the more highly expressed a gene is, the more accurately it can be measured. And after a square root transform, measurements contribute approximately according to how accurately they are measured. And the Pearson residuals instead identifies non-uniformly expressed genes, and lets measurements from those genes contribute more than they would after a square root transform. And they can be more effective than simple transform in separating out subtypes of cells. So here are some papers that are relevant to what I talked about. If you want to do some further reading, I also put links to these papers in the description of the video. And that's all I have, and thanks so much.